Hello, it's been a while since we've watched a video, so why not put one out now? Okay, so today we're going to be talking about protein synthesis. So right now, what you currently know about DNA is basically how it's made up, the shape of it, the nucleotide and what consists of a nucleotide, and how those pair up together. So, the A's with the T's and the G's with the C's, or the cat in the garage, the tree, apple in the tree. So that's what you know right now. We're going to take that idea, so now you know about the structure of DNA, we are now going to focus on the central dogma, because the central dogma talks about how DNA goes goes to RNA, which then goes to making proteins. And that's the whole big deal here. We need to make proteins. So the process in which cells make proteins is called protein synthesis. Synthesis meaning to make up. And there are two ways this is actually done, or there's two processes that go into this. One called transcription and one called translation. Because DNA cannot just go right into making RNA. There has to be a little bit of a process to it. The same with RNA. Can't just turn into a protein. There's a little bit of a process into that as well. So I'm going to go into each one of these in this video. Please don't freak out. I understand there is a lot of information in the last video and there will be in this video. Don't freak out. We took a lot of time learning about the DNA structure and you built a model. I'm going to spend just as much time on this. Do not think that I'm just going to let you try and figure it out by yourself. We have been going through things baby step at a time. The very basic process that we'll be talking about, like I said, is transcription and translation. Transcription, basically it takes the DNA, uses the DNA as a template to make an RNA molecule. From there, that RNA leaves and then it goes into the cytoplasm, and that's where translation occurs. So we're going to be focusing on, in the next few slides, we're going to be talking about transcription first. And this is basically talking about DNA converting into RNA. After this video, you should be able to give an overview of transcription, describe the genetic code, and explain how translation occurs. I don't know why there's two dots there. There really shouldn't be. Okay, so like I said, we're going to be talking about transcription first. This is the first portion of protein synthesis. So what we're talking about is we're going from DNA to RNA. That's the first part of the central dogma. Remember, the second part is now RNA to proteins, but we're not worried about that right now. The first portion is called transcription. It's the transfer of genetic instruction in DNA to make an mRNA, which is messenger RNA. Do you remember the role of messenger RNA? If you don't, look back in your notes, try to figure it out. So the basic gist of this is that during transcription, a strand of messenger RNA is made that is complementary to a strand of DNA. Now, one thing I want you to remember is that what nitrogen base does RNA have that DNA does not? Which one of the nitrogen bases is replaced? Did you think about it? So if you remember, the T, thymine, is not found in RNA. So that means when we are making this complementary strand of RNA, the T's are all going to be replaced with U. So that means A is going to bind with U on the RNA strand. Now, transcription happens in three basic ways, or there are three components or steps that make up transcription, which is initiation, elongation, and termination. Basically, we're starting, we're making something longer, and we're ending. Those are the three basic steps. And it's not that simple because this is biology, and why would anything ever be simple? I am going to talk about those steps in class, and I'm not going to try and speak it and write it all right here. But like I said, you should know initiation, elongation, termination. Those are the three steps. Make sure you have room at the end of the slide. As you can tell, I don't even have a lot written on this slide. I do plan on adding those three steps and their details behind them. I'm going to make them a little bit more reader friendly so that you're not like freaking out. So once the DNA has been, remember we used the DNA, one strand of DNA, to make the mRNA. Basically, mRNA is like another partner, and that mRNA or that RNA is responsible for taking the information from DNA to get out of the nucleus and go into the cytoplasm to help make this protein. Now, that's great and dandy, but unfortunately, that mRNA is not ready for protein synthesis. So there's a couple of things that must be done. So the new mRNA made in that should say transcription, not translation. Let me go ahead and change that right now so you don't even write that in your notes. Transcription. So the new mRNA made in transcription is not ready for the next step, which is translation. It has to go through more processing. So it's basically like the editing process. So let's say I'm making this video right now, and this is the first time I'm making this. I go through, after it's all done, ended, I go through and I edit. I take out parts I don't like. I add in maybe a couple other notes, or if you hear like little pauses and then me talking differently, that's generally when I've edited through. So that's my process of processing my video. The same process has to go for mRNA because like in any process or any organism, there are mistakes that can be made. And every single organism wants to go back through and edit to make sure that mutations are not occurred. There are 3 ways in which this processing of mRNA is done. It's called splicing, editing, or polyadenylation. And like I said, this is just a modifying process to make sure the mRNA is actually good. It has everything it needs. If for some reason that mRNA is not good or there's something wrong with it, that's when it's scrapped and we start over. So it's a great way for our bodies to have a check and balance system. The next part I want to talk about is actually reading the genetic code. So the first thing you might be asking yourself is, what is the genetic code? The genetic code consists of the sequence of nitrogen bases A, C, G, and T. And remember, U if we are talking about RNA. And this is all what makes up that polynucleotide chain. And those codes are great, or those letters are wonderful because what they do is they form in threes. So let's say I have AUG, AGA, CUC, and I'll write that down. So AUG, AGA. So those are a couple of pieces of a genetic code. And each triplet of those nitrogen bases is what's called a codon. And those codons, they code for a specific type of amino acid. And if you remember, amino acids all together, a whole bunch of them make up a protein. So here we're slowly going from the idea of making that mRNA is read into some reason that breaks down the amino acids. Those amino acids, or not breaks down, but forms those amino acids. Those amino acids all together will make a certain type of protein. So it's a pretty impressive process. It's actually very unique. Big things right now I want you to remember is that each triplet of nitrogen bases makes up a codon. So AUG, AGA, CUC, UGA, any kind of combination you can come up with. There is a table I'm going to show you in class. I do not expect you to memorize it, but I do expect you to know how to use it on the types of amino acids that are made based off of the different nitrogen bases. So for example, the nitrogen base UUU makes amino acid phenylalanine compared to CUU, which makes leucine. So very different combinations will make different amino acids. Now, some combinations will make the same type of amino acid, but the combination amount that you have can make a variety of proteins. So that's what's really beautiful is we don't even have to have a whole bunch of letters to make this beautiful organism that is us. So like I said, we are reading that genetic code. So when a specific codon is formed, it tells the RNA it's ready for translation. So here's what I'm going with. Remember, we were talking about mRNA, and we have to do that editing process. So splicing, polyadenylation, and editing. Now, once there's a specific codon that's found by a special enzyme, this is what tells the cell and tells that enzyme, okay, this is ready for translation. We can move on and we can now start making proteins. That codon is found, things start attaching, and we have some special structures that go on. So the basic breakdown is that mRNA molecule is read codon by codon until there's a stop codon reached. And I'm going to tell you what that stop codon means later. I know right now you're just like, what is going on and what is she saying? I promise you I'm going to talk about this much slower in class. So please, please, please just try and follow along. Write your notes down. I will have tons of notes added in class, so don't worry. But right now, the basics of what I've talked about so far. In transcription, we've gone from DNA and we've made mRNA, so messenger RNA. This all occurs in the nucleus. From there, that messenger RNA has been fixed up, made to look very pretty, and it has a special code on it. That special code then tells the molecule and the cell, okay, we are now ready to go on and make proteins, because that's the end goal. We want to go from DNA to protein. Very end goal. So now what's that look like? All right, I kind of already said this in the last slide. I think I kind of mixed some slides up or something. Either way, the genetic code, like I said, these letters combined in group sets of 3, they form code words called codons. Yeah, I'm pretty sure this was supposed to be the slide before the other one. I don't know what happened. Either way, those codons are read on the mRNA sequence, and once a specific codon is found, we either start or we stop, depending on the situation. All right, now let's talk about translation. We talked about transcription, which is the first part of making protein synthesis, or making proteins. Now the second part is all RNA to proteins. Now remember, in transcription, we made what type of RNA? The first letter is an M. So messenger RNA. Now we're in that messenger RNA sequence and we want to make proteins. So this is the second part of the central dogma. It is a process in which the genetic code in mRNA is read to make a protein. So whatever the DNA deemed necessary is the instructions that it gave to the messenger RNA. So remember, messenger RNA is the complementary pair of the DNA. So then that's going to be read, and then whatever sequence of codons that's found, that's what's going to indicate whatever protein is made. So you can have a small protein or you can have a very large protein. It depends on the case. Transcription is what happens in the nucleus. As mRNA proceeds, it moves out of the nucleus, it moves to a ribosome, which is found in the cytoplasm. Now we have another type of RNA that's going to come in. So after messenger RNA leaves the nucleus and moves to a ribosome, which consists of ribosomal RNA and proteins. The ribosome reads the sequence of codons in the messenger RNA. Molecules of tRNA, which is translation RNA, bring amino acids to the ribosome in the correct sequence. So for example, if there's a sequence that says U, that tRNA is going to say, okay, I'm going to translate this, I'm reading this, and you are telling me that you want to create that specific amino acid. If the sequence says ACG, that's a different amino acid. So then the tRNA is going to say, okay, I'm translating this. This is the amino acid you want me to make. Those amino acids will all bunch up and pair and pair and pair, and eventually you have what's known as a protein. So tRNA has like a really nice way of doing this. It does this by pairing an anticodon to the actual codon. So like I said, the mRNA has codons. And like if it's UUU on the mRNA, the tRNA molecule comes up with the anticodon. So it translates it. If it's UUU, what's the only nitrogen base that binds with UUU? I hope you said AAA. So that's the anticodon that goes with UUU. So that's what tells the tRNA molecule, this is what I need to make, the opposite or the complementary pair. This is a very, very basic breakdown of transcription and translation. I completely understand if you are overwhelmed and if you feel incredibly, maybe like a little bit insecure about this information. It's okay. It's a long process and it's incredibly overwhelming if you let it get that way. It's okay to feel overwhelmed. It's not okay to give up because you just don't understand. We are going to go through this in class. I'm going to sit and explain it, and I will take as much time as you need. Please make sure you are asking questions, doing what you need to do to make sure you understand the information. We will probably have a quiz on this and later in probably sometime next week, so please make sure that you're keeping track with everything we're doing. Again, if you have any questions, ask me on Edpuzzle, please.